So Revati is also joined. I think she has some issues with her internet, so she might not be.
Sure, sure.
So it started. It's, I think you can check it out on YouTube.
No, I don't have to check now. It's okay.
Yeah. Okay. So I'll start around 6. Let's just wait for 50 more minutes. That's fine.
Sure, sure.
I'll be back just 1 minute.
Sure. So you guys are working from your institute, I see.
Yeah.
Expecting your return to home soon, I guess.
Sorry, oops, I couldn't hear you.
I mean, there is a possibility of partial lockdowns and lockdowns happening again.
Yeah,
So,
that's the vibe. Yeah, it's a little tricky. So I'll start off, Ipsa. I'll just introduce.
sure, sure, sure.
Yeah. So good evening, everyone. I am Spurti from Indian Institute of Science Education Research, Tirupati. And on behalf of our team, I would like to welcome all of you to today's talk organized by Inventa. So, Inventa is an inter institute science magazine and it is the first of its kind, being a student led venture from the students of seven Indian Institute of Science Education and Research, the Indian Institute of Science, the Center of Excellence in Basic Sciences, and the National Institute of Science Education and Research. As an initiative aimed at spreading awareness and information about science to students and professionals alike, it is fitting that we hold space for a discussion about science communication itself, and that too through alternative means besides academic settings. So, it is a great pleasure for me to introduce our speaker today, uh, Dr. Ipsa Jain, who is going to talk to us about drawing science. So, she is a scientist and a molecular oncologist by training and is now a science illustrator and a communicator. She has her own freelancing outfit and works with scientists, educators, and institutes to develop visuals for audiences from diverse backgrounds. She also makes independent designs for SciComs, and in addition to all of this, her work also initiates conversations around sci art and women in science. We will host an interactive session after the talk, and we encourage participation from the audience. Please join me in welcoming Dr. Ipsa Jain.
Thank you so much, Spurti. A pleasure to be here. I always uh, accept all the invitations that come for talks from wherever they are. I'll begin now, I guess, the discussion on drawing science. Sorry. Please give me just audio feedback if you're able to see my screen.
It's visible.
Okay, excellent. So, good evening, everyone. Thanks for being here on a Saturday evening. I suppose there isn't much opportunity to socialize, so Zoom is one way. These talks are one way. Uh, so, uh, so I'm somebody, like Spurti introduced, somebody who spent a lot of their time trying to train themselves as a scientist, and I like to think that I'm still scientist at my core. But the tools I use have switched, so now I use a pencil or a brush instead of using the pipette. And as a proof of my credibility, I'm just going to give you a glimpse of some of my work. Um, in this session, I would like to impress upon you the relevance of visuals in science overall. So I'll start by saying that as humans, we are curious. We want to see what's around us and observe what's happening. And there's no reason for it except for the hell of it. We just want to do it. And we want to look at things, we want to touch them, taste them, smell them. And this practice of us as humans has even changed the way the nerves are concentrated within our body and the way the muscles are tuned within our body. So we are sort of proficient in doing really fine tasks with our hands. And knowledge making and hence science as a process is both a function of the mind and our hand together. And that's where all the material and knowledge production happens. And so science is an act of mind and hand. So I'll just take you through some visuals in science and impress upon you that how visuals have been part of the scientific discovery as well as science dissemination throughout the practice itself. So this is detailed in anatomical drawings by Vesalius, Euclidean diagrams by Copernicus, microscopic drawings by Leeuwenhoek, paths of light by Newton, arrangements of instruments and tables by Lavoisier, relationship between different specimens by Haeckel. And after a point, so all of the examples that I shared earlier were based on hand drawing based method, and after a point there was technological advancement and we could take photographs, and not just photographs to the usual scale, we could even hook them up with telescopes and microscopes to take pictures of the sun here. And we had, sorry?
Sorry to interrupt.
Yeah.
Can you make this like full screen on your slides?
It is full screen on my system. Are you not able to see
Okay.
it properly?
Yeah, can you just present it once more?
Sure, I can do that. Can you see the whole thing?
Yeah. Thank you.
You're welcome. Yeah, so after a point, we also had advancement in inventions of other image producing equipments like the X-ray based analyzer here. And something changed with Mudrij. He changed culture, and he also changed science with the discovery of sequential imaging. And after a point, we could hook up these recorders to microscopes. And this was like one of the first few visions of cells moving and touching each other. You could see the vesicles within the cells moving, the membranes ruffling. So much drama could be seen. And with more advancement in technology, we could now look at images in far more intricate detail and get access to information that wasn't, available before. And some of these works look like work of arts themselves. In this image, the rainbow, you could sort of feel like you understand each neuron and how it's connected to the next one. So much information adds so much beauty. And with such advancement, has been such a huge influx of the way image has been made in the surface of science. But with all this advancement, is the role of hand lost? Is drawing lost? I might be, of course, biased, but I think not. And let me try to uh, convince you. So, in his notebook, Darwin starts with a phrase, I think, and ends not in a sentence, but in an illustration. A tree that represents the key concepts diversity, descent, and extinction, the great tree of life. So, visual displays are distinctly involved in scientific communication and in the very construction of scientific facts. Observations are the fundamental base of all research methods. The beautiful images in Maria Sibylla Merian's Metamorphosis, Insectorium Surinarium, the work is the product of meticulous work where she spent time looking at each specimen, making specific notes, making specific drawings. And when she put all of these together to make this composition where different elements were all present in one final resulting image, she changed very little in terms of how the animal looked or the plant looked or behaved or their interaction with each other. Seeing, in some respects, is an art which must be learned. In his preface to Micrographia, Robert Hooke wrote that all that was required to study nature was a sincere hand, a faithful eye to examine and to record the things themselves as they appear. In this passage, Hooke implies with his words that with his words and pictures, he acted only as a servant to the microscope. And his illustration of an ant is made of a scale which is unusual, much larger than its actual size, and it highlights insects, spiky limbs, brittle structures, and puts it against a stark white background, thereby transforming it into something that looks like an alien. And when he built these images, he looked at these specimens multiple times from multiple point of views, at multiple magnifications, and so on. So vision is not a mechanical recording of elements, but rather an apprehension of significant structural patterns. In the more recent time, an example from the more recent times. So perhaps some of you have used or know of the Drosophila atlas. Similarly, there are a set of drawings which are used for staging of the zebrafish embryo. In 2017, a scientific illustrator worked with a team of this is Xenopus, not zebrafish, Xenopus biologists to redraw and redefine these stages. And her process involved looking at the specimen directly through the microscope, drawing several sketches from multiple specimens and multiple times, and then she averaged all these drawings into this one final drawing and added all these details. And what happened for her during that process, that she was able to identify, notice, and document and depict details on optical lobe development that had been missed by the previous set of drawings made earlier. So this hopefully convinces you that drawing is an important part of making observation. Now the next thing I would like you to do while you're with me is to imagine how a cell looks. Are you thinking of a cell? I asked some people in some other workshop to do just the same, to think about how a cell looks. And these are some of the representative responses I got. You can see a lot of these images are the circle within a circle, a flat cross section of the cell. I and this, in some sense, is reminiscent of the kind of structures we see in our textbooks around us. So these students were students who currently engage with biologists or biologists in training, much like yourself. And when I asked some of the non-practicing uh, people about how they remember the cell, this is what they do. These people are right now architects and lawyers, and last time they engaged with biology was probably in 10th standard. And you see that here also is a circle or a dot within a circle. So the way we learn images during our early education influences what we remember. And the biggest, so before I go on to making my point, I'll just show you some of the images from the history of cell biology, which are also prevalent in the way we look at cells or think of cells. This is from the time of cell theory, this is from the time of cell division, this is the modern way of looking at cells through highly specific staining. These are sort of icons that you might find online, and this is a 3D icon. This is a comic representation. And another question is that when you put them all together, what do we learn about the cell from each of these images? This is being done on YouTube, so we can't necessarily make it interactive. But I'll give you a minute to just spend some time to look at these images carefully and think about what are the differences in terms of what you learn about the cell from these images. All right. So let's think of some properties like cell types. So only few of these images, the first one and the last one, consider different types of cells. Let's think about volume. So while it's not clear at the scale here, the picture 1, picture 2, picture 4, and picture 5 here on this slide consider the volume of uh, the cell. If we go back to the previous slide, the third and the fourth images are completely flattened. Right. Then we think about the dynamic nature. Are these cells breathing? Are they doing something? So again, if you think about the second image and the last image, there is some change there. In the second image, you see that there is a difference in the way the chromatin is arranged within the nucleus. In fifth one, you have the sense of cells moving and ruffling and changing. So this is where you feel the dynamic energy of the cellular processes. Some of these, especially the last one, takes you through some cellular process. You see that there is an indication of phagocytosis there. So let's not cover all of them, but you see that the different kinds of images view different sets of information and you need basically a diversity of images in your memory to remember different parts or different properties or different aspects of the cell here. And this could be, this idea that you need a lot of visual stimuli, different kinds of visual references to make a cohesive picture could be true for any other subject. So I, being a cell biologist by training, I of course am very interested in how I think about cells and how other people think about cells. So I wanted to challenge this status quo where we think of cell as this sort of a sunny side up, where the nucleus is within the center and there is a nice round shape around it. So the first thing I wanted to change, the first idea I wanted to present was that the cell is not a flattened object but a 3D entity. So to do this, I devised what I think clever metaphor. What I did was that I sliced the familiar object in orange, and then I sliced a cell, tetrahymena in this case, and then once I got a piece of orange, then you got a piece of tetrahymena. You look at a piece of orange, and you look at a piece of tetrahymena. And what happened with this metaphor was that I published this as a zine, and I was able to present it to different kinds of audiences. And whether they were trained in sciences or not trained in sciences, they could see relationships between organelles, distribution of organelles across the depth of the cell, the polarity of the cell, and people who were not trained in sciences could also build these relationships despite the fact that they did not know that this organelle is called mitochondria or that this is a cilia. A lot of people were able to tell that between the placement of the mitochondria and cilia, for example, that there is a very close relationship. So even with just the images and removal of jargon, it's possible to convey ideas through drawings. Say that we see, but we do not see. We use our eyes, but our gaze is glancing, frivolously considering its object. We see the signs, but not their meanings. We are not blinded, but we have blinders. So in 1968, Waddington writes, science is more than a collection of conceptual or practical results. It's also an activity, and its practice involves, as a very important part, the exercise of the faculties of insightful perception of the natural phenomena and of imaginative creation of new concepts. This is the part I'd like to emphasize on imaginative creation of the new concepts. So the image on the left is a top view of Waddington's epigenetic landscape, where the path followed by the ball represents a developmental trajectory. The valley represents different alternative paths to depict different developmental potentials. For example, stem cell staying the stem cell or differentiating at the next cell division. On the right is the undersurface of this landscape, where these pegs, which represents genes, are interconnected with each other and are also controlling this mesh, and hence the topography of the surface where the ball can roll through. And these images do something, which is that they stimulate thought. They provoke you to think about this concept. And the way he wrote it, it was to loosen the joints of the scientist's imagination, creating a space for associative play required to introduce new concepts in theory formation. However, there are, of course, criticisms to this theory, one being that the ball stays as the ball throughout its journey. And artist Gemma Anderson, a drawing artist Gemma Anderson, wanted to change that. So she collaborated with scientists and superimposed this series of transformations on this Waddington's landscape. So she had previously created this series of drawings, which she calls morphological transformation, where you see an organism developing from a starting point, like an egg or an embryo, and a metaphorical representation of that. And this image then just superimposes both of them. And this image then transcends above what Waddington was able to achieve. And Gemma in turn writes that creativity, the formation of new associations and concepts, requires playful abandoning of preconceived notions. To be creative, we must let our minds wander. She says hand drawing must not be allowed to disappear. And it's not just because of its use in scientific illustration, but because of its potential to form insight and understanding, and to share this understanding and to enhance creativity among scientists and artists alike. All right. And now what I'm going to do is share some short glance into some of my own work where I tried to use drawing as a way to explore science. Uh, this work was done at INSTEM and Institute Curie. Um, so Renaud, my collaborator here, said that drawing enables the creations of images that are more abstract than photographs, but have more depth and are more accurate than schematics. Drawings can provide special visualizations that could never be photographed or easily schematized. So we wanted to understand how, okay, this place, excellent. So we wanted to understand how the cargo moves inside the cell. So here what's happening is that melanin filled in pouches is moving from outside to the center of the cell. And this happens because of this molecular motor called dynein. You don't need to understand a lot about dynein to understand what I will just share with you. So we wanted to consider how these molecules work together, how do they move, and so on. So I started by looking at the structure of this molecule and sort of started sketching it out. I made some rough drawings and made some sort of niche drawings. And once I sort of started to understand what the form of this molecule was, I wanted to understand how the form changes, uh, when it goes through a walking cycle. Uh, so, and this molecule does not work alone. It has like several other molecules that are attached to it, and then of course the final cargo that is tethered and that moves along when this motor is it works. So what I did was I read a lot of literature. I first was trying to look at the structural aspects of this molecule, this super complex. How does it look? How are the things in relationships to each other? And once I was sort of able to find some answers, some of course still remain unanswered. I then could ask next kinds of questions, which were more related to its function. So I was able to move from structure to function by just the practice of drawing itself. I was like, okay, this moves this way. Then how does this change how it interacts with the next thing? So these kind of questions were possible because I was trying to summarize the whole thing with the drawing. And finally, when I was able to identify some pieces of this jigsaw puzzle, this is the final drawing that we came up with. And this drawing is cool, not only because it summarizes all that I could find, but it also summarizes stuff that I could not find. So the way I have used my pencil, the intensity of the pencil, or the use of lines is indicated or indicative of what we know for certain and things we don't know for certain. Once I was done looking at the structural aspects of it, I wanted to understand how it works. So you don't have to look at this data and feel intimidated. The only point I'm trying to say here that there was a lot of data available, and through that, on the right, you see, I was trying to figure out like where permutations and combinations. So what we understood about this molecule was that it could walk in uneven steps. Like it doesn't walk necessarily like how we walk, and it could take uneven steps. It could go from covering 8 nanometers to 18 nanometers or so on. The numbers don't really matter here. So while I was trying to understand this, a new structure came in, and that gave me far more clues as to what's the starting point of this work. Does it start at the same point, or are the two molecules at a gap with respect to each other when they start working? So this is what the structure did for me. And after that, I was able to extrapolate and figure out these various market permutations and combinations. So what I'm going to do now is just, we don't have to go into the scientific detail of this whole entire process, but what I'll do is I'll just play these, different walks in quick succession, and hopefully you will be able to appreciate that different walks meant that different distances were covered. So this line that's on the left side tells you how much distance did the molecule cover, and you see that with different permutations and combinations that was different. So with this whole exercise of trying to put the structure, function, and the walk together, we were able to identify far more new questions that are perhaps being answered by the scientific community today. How when the motor works, how does the cargo move? Does the cargo move when both the participants in the motor complex have moved, or can the pull of the one molecule itself pull the cargo also? So these kind of questions came up because of this exercise of putting together this drawing. All right. So now I want to give you an exercise which hopefully will be useful to you. Um, I want you to take a look at this picture, spend some time with it, and the next thing I would like you to do is try and imagine what happens to this next. And the next could be 1 second, 1 minute, 1 century, either in the forward direction or in the previous, a back in time direction. And why I'm asking you to do this is because during the, like Jim Anderson puts it, drawing is a playful exercise, and it's free from the pragmatic constraints of experimental feasibility. You can draw what you imagine, and from that imagination can come up hypotheses that haven't been explored. So a hypothesis may be visualized, worth pursuing may be visualized verbally or visually, and the latter may be described rather than depicted. Um, and I want to end by saying that while no one would claim that images are integral to all scientific thinking, it is hopefully indisputable that scientific thinking, sometimes of the highest order, occurs in the absence of words. And when probing into the unknown, drawing is an ideal tool because drawing is thinking, pointing. It makes things present and puts them at a distance. It is a mode of inquiry. Drawing can represent seen and present the unseen, the known and the unknown. Thank you for your patient listening. I'm hoping that there will be questions uh, after this, and I want to thank my labs where a lot of this work happened. I want to say that I'm easily accessible online. You can find more about my work on my website and social media. Everywhere I am present as Ipsa. Thank you.
Thank you, Ipsa, for the really wonderful talk. So I have a few questions, and I'm hoping the audience will generate a few more to keep the discussion going on.
Okay.
So I would just like everyone to post their questions on the chat box so I can read it out later. And maybe in the end, we can actually let you guys speak if it's a really cool question. Okay. So my first question, Ipsa, is
Yeah.
so actually, what advice do you have for the audience who want to pursue the artistic side of science?
Okay. So first advice is to do the activity that I just asked you to do. The thing to, I mean, how I started was literally with a pencil and paper, and that's still the tool that I use. Um, so I think the first thing one has to overcome is some sort of fear-based inhibition. And I think pencil and paper are a very forgiving medium. Don't make it for anyone else. Don't make it for your friend's blog or whatever, or your own blog. Just make it for yourself and practice uh, until you start feeling comfortable with using the tools. And the next thing would be to talk to people who are doing this kind of work, ask them a lot of questions, read about their work. That's what I did. Like when I started into science illustration and science art, I interviewed and blogged about it a lot. I interviewed several science communicators, several science artists, and that's an exercise that I tend to repeat after 2, 3 years, so that it sort of also is a part of how I think about my growth. So talk to people, practice, and yeah, that's the two advice that I can give right now.
Thank you. And so one more question is, like, which is your favorite art form for depicting a scientific phenomenon?
Sorry, Papri. I don't know if I have a favorite art form, but I can probably say that watercolor and pencil are some of my favorite mediums. So maybe that. But when I think about what medium to use, there are several factors that guide me. That could be whether it's something that has to go online in the journal, then of course it has to be, done using a digital software. But if it's something I'm doing for my
Hmm.
own personal sort of work, then it's pen, paper, and watercolor all the way, really, unless the subject necessarily demands it. If I'm making something which is quite geometric and has too many specific dimensions, then I might go for a digital tool.
So we have a few questions from the audience.
Sure.
So Vishnu has a question. So, do you think that UG students should have separate courses for artwork and design since science is heavily dependent on these?
So I mean, I would always, of course, advocate for art and design courses, and I wouldn't necessarily say that it's because you can use it in illustration or something like that. It's just because also it's a different way of thinking and approaching a problem. So I think design thinking also helps you understand how to solve problems in a very different way, and art is a more freer space where it doesn't even like every individual has their own process, and how you approach an art piece or how you respond to an art piece would also reflect in some way how you respond to a PCR not working in the lab or a code not working in the lab, or so it's both a way of building your cognitive abilities, testing yourself as a person in some ways, and also, of course, the very direct sort of output in terms of science
Yeah.
communication or making diagrams for your work. Yeah, if that answers what you meant to ask. Okay, is it my internet or um, is this Ushti's internet? Can somebody hear on the
Uh,
call?
yes. And uh, sorry, is there a delay?
Yeah, yeah, I
Like, is
couldn't
there a
hear
lag
you
from my side?
either side, but yeah, I couldn't hear you for a bit. Yeah. Can you repeat your question, the next question you asked, please?
Yeah, 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 yeah. So basically, the question is, can pencil art be replaced by digital art?
So that's a question that I get asked often, and I think it's a lot to do with personal preferences. There are people who I think would never leave pencil, and there are people who are much more comfortable with vectors or Photoshop or whatever. So I think it's a very personal choice. Of course, for publication, it might help if you are somebody who can prepare the files digitally also. Uh, but I think as a thinking tool, I still think that pencil and paper are far more organic and far more giving and far more useful than Photoshop. I think ideation still happens on paper for a lot of people who end up working digitally as well.
Yeah, like I think that's the place to use a lot of digital art.
Yeah.
So we have another question.
Yeah.
So how can scientists actually groom their art, like uh, artistic side for promoting their research to public, like research to public, basically? So do you suggest us to take drawing classes and
Can you repeat your question? I actually lost you for a second. I'm not sure if I understood the question clearly. Sorry about this internet issue.
Is there a lag again? I'm sorry. Okay, so basically, my question was, so since not all of us actually know how to draw, and since we are going towards science in academia, like few of us, so how do you like suggest us to take drawing classes? Like, how do we actually, you know, indulge or like, promote our artistic side?
Okay.
Like, if
I
we
mean,
are
of course,
not that good of an artist.
okay. So, I mean, when I take workshops, I always tell people who are in the workshops that, um, like, art doesn't have to be about, your end product being uh, beautiful enough or something that you can put up on social media. Like, that has to be a personal experience, and it's more for making you feel involved and engaged about the idea that you're thinking about. So when you're making any work, I mean, it doesn't even have to be drawing or painting. I mean, whether it's you're dancing or you're singing or you're writing a poem or anything that you want to do, what happens during that process is that you are engaging with whatever is your idea for a long time. You are contemplating and calibrating it. And for some, it is an iterative process. At least for me, it is an iterative process. So, what matters is how much time and thought you spend with your idea rather than what's the end product. Like the end product doesn't have to be pretty. That's not a requirement to create art.
Okay, thank you. So we have a few questions from YouTube. So
Sure.
so we have a question. So I am often get the idea that I am not being productive enough when I am drawing. It would be great to hear your thoughts about those feelings.
I mean, so I think this is something that academia does to you also, like imposter syndrome. Hello, like I think you enter into an institute and it's like hello imposter syndrome. It's like some mandir ki ghanti or something, and uh, I think we should not put that pressure on ourselves to be productive. Like I understand that can happen sometimes when you're working with a client or a collaborator and there is a timeline, and then it might be important for you to sort of feel the need to be productive, but I don't think productivity should be a criteria. Practice should be like if so. Practice is where you spend some time almost every day and work on something. But again, that work doesn't have to be lead to output. So like when you use the word productivity, it is often about an end result and output. So that's not necessarily a requirement. It's the time you spend to hone your skills or develop your ideas. That's also part of your growth as an artist. So I don't think you should punish yourself for being not productive. That's that's a PhD course in some lab speaking. That shouldn't be you doing to yourself with yourself.
Yeah, so we have another question from same person. So they're wondering about the career options in this path.
I am also freelancing. I don't have a job, so but I'm hoping that there'll be a job for you tomorrow. But uh, I would say, like at this point of time, I think you should just diversify the kind of things you do or would want to do, and use that as a way to generate income and stability in your life. Or you could always have a day job and do this as a night job if that's something that you think you can sustain. Um, uh, it's challenging in India right now. I have to admit. I mean, we do have some space for writers, but for illustrators right now, we don't have that kind of space. But I think there is uh, some work to go around, and you can always create your own work and find ways to sell it, put it out there, and sell it in some way. I mean, I do that also. So yeah. That's the only suggestion. I can only give you that advice and then hope for you.
Thank you for that insight. It was required. So we have another question. So So, a person is asking, so Malvika is asking, is scribbling considered part of expressing science? I'm not sure what that considers scribbling as, but yeah, what is your opinion?
Okay, so it depends. So if it's for you or a very small cohort of people, it can certainly have meaning. Or if you're thinking of it from a completely artistic perspective, where the practice itself is what you want to sort of convey to your audience. But if you're thinking of it as like science communication, that may not work. But if you're thinking of it as an artistic
Okay,
practice, then of course you have the freedom to consider anything you want as an artistic piece. Yeah. This is something my friend Gamana said to me, and I don't know if he remembers also whether he said this to me or not, but he said that like when we make art, like when you're thinking of something as an art, then you are making with a certain intention. And when the audience is interpreting it, the audience is interpreting it based on their own context and their education about how to look at visuals. And I think the success comes from if the gap between what the artist intended and what the audience understood is less. I mean, it will never necessarily be the same because lived experience and the context that you're coming is very different. But if you are able to convey what you meant to convey, then you did a good job as an artist. But if you weren't, then maybe there's a way to go. And like, I think a different set of artists might not necessarily agree with this kind of statement, and I can understand why. But I think when we think of it in terms of science art, that might certainly need to be deliberated upon.
okay. So we have another question. So, okay, so this is pretty interesting. So the brain is attracted towards art than literature. So we prefer seeing more pictures than words. So is that something to do with the evolution of our ancestors?
I wouldn't know the answer really, to be honest. I haven't paid this enough attention, but I am not sure I agree with the premise in itself that we are more attracted to visuals than words. I think that they're just different set of people. There's different set of people who like words, and there are different set of people who like images, and maybe there are people who like both or a different ratio of both. Uh, yeah, but I wouldn't know an honest answer to your question. Yeah, sorry about that.
Thank you for answering. So, yeah, so one question from my side is how has having a PhD impacted your career?
Um, in several ways, I would say. So one of the first is especially when I try to make a living. So when I try to make a living, that means I work with scientists to sort of create illustrations for their websites, like grants or papers or some things like that. And when you try to do something like that, it really helps that you have a PhD because if you're somebody who does not know science at that level, then I mean, I would say scientific training, not say PhD degree. I mean, uh, as long as you have the ability to understand their language, like scientists speak in some sense in a very esoteric language. So when they talk to me about an experiment or an observation, they probably don't have to spend as much time explaining to me about what a DNA looks like or what the soil looks like or something like that, just because I come with an understanding of those things. But if they were working with an illustrator who was from the outside of a scientific community, who probably is better at making illustrations also, but they would probably have to spend a lot more time trying to convey what they intend to convey. And I think so that's the one reason scientific training certainly helped. Then the second thing, of course, in terms of exposure and understanding, I myself understood a lot about science and scientific method and images in science during my own scientific training. Then the third thing it really helped in towards is networking. So because I mean, but that would still be true for people like you who are already part of a good institute. So being part of ISC or INSTEM campus, what it did for me was now I was able to interact with a lot of scientists from across the country or even across the globe because of being in these places. and tell them about my work and share my intentions, so that in turn got me some projects which maybe I wouldn't have been able to procure if I was living in my own way. Yeah.
Huh. Thank you for that. And I have one last question. So,
Sure.
how did you actually get from art as a medium for, how did you get into art as a medium for science communication?
Okay. I mean, the journey honestly really started because I, towards the end of my PhD, figured out that this is not what I wanted to do with life anymore. I was done with the PCR business and cloning business, so I had to spend some time thinking about what is it that I would want to do, and talking to people about science came as an sort of an obviously easy answer. But then I had, at that point of time, no confidence in my writing abilities or speaking abilities. Not that I used to know draw a lot, but I still somehow felt confident about the idea of drawing, and that's how it started. And but the idea of what science illustration means, or the practice of science art means, that in itself has evolved through time for me personally. When I started my journey, it was all about making representational drawings. I would take a specimen, draw the specimen, and that would be my scientific illustration. Um, or make some summaries, sort of schematic, and that would be my work. And I think it was after I started looking at a lot of people's work, understanding how they think about art, looking a bit at art history itself as well, and talking to all the science communicators that I did, that it shaped and changed the way I approach this idea. So now science art is more about also either using the scientific methods as a part of the artistic process, or using the artistic process to tell stories of science. So or even a broader definition, a loosely ill-defined, jelly-like morph, where there are several possibilities of how one approaches science or how one approaches art. Um, so yeah, so the and science communication is just like one small bubble within that whole jelly, or science illustration in itself is also a very small bubble within that huge jelly.
Thank you, Ipsa,
You're welcome.
for answering all our questions and for giving us this interesting talk.
You're
So,
welcome.
I would like to conclude this session here. And I would like to present the vote of thanks on behalf of the editorial board of Inventa. It is an honor and privilege for us to have Dr. Ipsa Jain here today, and we are immensely grateful for that. It is a delight to attend your talks, and we discussed so many topics today. I'm sure each of us found something to think about, particularly concerning art and the wonder of mind and the ease of science communication. So, Inventa was first discussed as an idea way back in July 2020, and the transition from ideation to actualization has been made possible with countless people's timely aid and cooperation. So, we are grateful to the directors and faculty members of all the participating institutes, the ICERs, IISc, NICER, and CEBS, for extending their support to our fledgling initiative. So we thanked Professor Satyamurthy and the editorial team at Resonance for featuring us in their January 2021 issue. We are deeply thankful for the Inventa team and all those who volunteered to send in their contributions to kickstart this initiative. And we hope to see more such contributors for the next edition. You can go ahead to our website to download the magazine and find some more information about this year's edition and the people behind it. Thank you all for attending the talk, and we hope you enjoy this edition. Once again, I
Thanks
would
for
like to
having me.
thank you, Ipsa.
Thanks for having me here. It was fun. Thank you so much. Good luck with the magazine. All right.
Thank you.
Bye bye.
Bye.